My short mobility or short-term scientific mission, my very nice stay at the dendrochronology lab of the province of Baden-Württemberg, which is in Germany, in a very nice town called Hemmenhofen. But before that, I would say a few things about the STSMs. What are they? What is this format? What is that platform? Who is it for? It's basically for all of us. It's not only for early career researchers or people from ITC countries or whatever other requirements COST may have for the other platforms. So basically, this type of mobility is literally for everyone. And it works in two directions. You could go and visit an institution which would host you, but you could also offer your institution, your lab or your university, your affiliation as a host institution to someone who would like to visit. So this is really one of the very nice platforms of the COST. And here I've put a small tiny screenshot of the definition that the COST has put on what is an STSM. So basically the key words are, well, these are exchange visits, obviously. And the key things are to share techniques and gain skills. So I think this is really the best combination that would describe such type of mobility, because this is not like the Erasmus days, for example, where you could have a full semester or 2 semesters somewhere. This is something shorter. So in a way, it should be really more focused and kind of to the point. You have to know why are you visiting your host institution, and obviously which are the benefits you could gain from that. And of course, since the whole COST thing is a networking mechanism, you have to think further. So there should be also like an added value of possible collaborations in the future. I mean, beyond the mobility, beyond the STSM, and perhaps beyond the COST action, which is, I think, the nicest thing in the whole COST. So obviously, there are a few things you need to know before the mobility. You need to identify your prospective host institution, maybe a person of interest, someone you would like to learn from, or maybe a lab which is famous with a certain technique you would like to learn, apply, or maybe practice. And once you identify your host institution, obviously there is a procedure of application, which is really not heavy. I mean, COST has made it very easy for us. And I think the key thing is you should really plan well, because if you have a week or 2 or 3, this is not a lot of time. So you should really plan well your activities, the things you're going to perform when you visit. And well, I think, as I said, one of the greatest things during the mobility is also the possibility to really think for the future, because they may be activities that you didn't plan, but they may emerge out of the conversation while you're sitting in the lab and perhaps performing your analysis or whatever you're doing as a visitor. So I think this is really a valuable thing to think about. And after the mobility, there is a little bit of bureaucracy. There are a few things that you need to keep in mind. There is a reporting procedure, and there may be also dissemination requirements like we do now. So you may be asked to deliver a short seminar, maybe write something for the social media. I mean, this is specific for each COST action. So this is it. More information could be found actually on the EU-PoTaRCh webpage. So you could read the details and obviously ask your specific questions. Just to check, do you hear me?
Yes.
Good, thank you. So there we go. I'm trying to change my slide. Does it work? I hate Teams. Okay, I think it works now.
Yes,
so thank you. The details of my mobility or the STSM. So my primary affiliation or the institution I come from as as it was stated in the beginning, is the Institute of Biodiversity and Ecosystem Research, where I work in the unit or the division of paleobotany and palynology. And specifically, what I'm interested in is called archaeobotany and anthracology. So I work with seeds and woods from archaeological contexts. So this is me, and the host institution is the Dendrochronology Lab of Baden-Württemberg, which is located in Hemmenhofen, very nice town at the shore of Lake Constance. And my host was Oliver Nelle. So what we did, we had two things that we wanted, two main lines of that mobility. And they were all dedicated to wood. So, and specifically to charcoal, wood charcoal. So wood charcoal is charred wood. And that charred wood originated from several archaeological sites. And that was a combination between a few charcoal production sites from Germany and an archaeological site from the late antiquity from Bulgaria, from the city center of Sofia, and part of the ancient Serdica town, which is the name that the city had during the antiquity. So what we did a lot of taxonomic work. For those of you who are not familiar with the charcoal analysis, so if I have to sum it up in simple words, each single piece of charred wood, each single fragment of piece of charcoal is being looked into 3 anatomical planes, because we want to, our first goal is to identify the species. So the difference between working with fresh wood and working with charred wood is, well, we're still looking for the 3 planes, but with charred wood, we have the possibility to actually manually break the fragment, because charred wood is fragile, and that makes things easy if you have a really good size of your fragment. But on the other side, it challenges the lab procedure when you have tiny fragments. So this is what you see here on the pictures left and right. You see tiny little black things, which are pieces of charcoal from different sites. And if you look carefully, you would see that each single box or each single plastic bag contains several pieces. So originally this was 1 fragment, which was broken into 2 or 3, because we needed to see the 3 anatomical planes. So we needed to look at the fragment transversally, tangentially, and also radially. So this was basically the core activity during my mobility, which I forgot to say, it lasted 2 weeks. Of course, that wasn't everything. Here, I've done a little summary again with the main things, but as you may imagine, if you're working side by side with someone in the lab, there are really lots of discussions and lots of things you could really talk about, discuss, and in my case, learn from your host. So as I said, the taxonomic bit was the big chunk of my STSM. We also discussed the values of charcoal production sites, which are, I would say that in the region I come from, archaeology is not really familiar with that types of, with these features, with that types of sites. Familiar by means of they're not that easy to be recognized and to be surveyed as archaeological sites. So we had really fruitful discussions on in that direction. We also talked about the classical and the modern survey strategies and procedures. For example, if you're hiking in the mountain, how would you think of, for example, charcoal production pit? And one way of that, or probably the classical way of that, is just looking into the map. So I know it sounds old school, but this is something you may have in your pocket. Of course, we talked about the potential of the LiDAR technology, which if it's done systematically on a certain region is literally a data bank. So LiDAR images could be something you could look at. Another activity, which was kind of a bonus, an extra activity, we did a little experimenting dendrochronological measurement on a piece of archaeological beach. We also discussed this peculiar species and the potential for dendrochronology. which was very interesting for me and made me read a lot after that. And of course, we pinpointed some ideas for future collaboration beyond the STSM, and obviously we talked about the conference participation and also co-authoring a paper together. And why mentoring is important, I decided to put that slide here just to remind all of you who are probably thinking of going on such mobility, working with someone you could really learn from. Well, mentoring is important because it's important, but also it's in the simple little things you may be missing in your daily practice. So here I've done a really nice anatomical illustration. I mean, it's not mine, the source is down the bottom, but I've put it here because here you see two slides of tangentially cut wood. So these are two deciduous species. You have willow to the right and you have poplar to the left. So that little bubbles that you see, you probably know what they are, but they're called the radial rays. So the red arrows are pointing the terminal cells of that rays. If you look at them carefully, you would see that there's some sort of homogeneity. So they're more or less looking alike. They're more or less similar. But the terminal cells of willow, which is the salix one, the one to the right, are slightly elongated. So they're a bit different than the rest. So that's why they're called heterogeneous, because they're not homogeneous, obviously. Vice versa, to the left, the red arrow points a terminal cell of a radio ray of poplar, which tends to be homogeneous. So their size, their shape, they're more or less like the rest of the cells in the ray. But as you could see in with the blue arrow, this is not always the case. So sometimes you could have both cases. You could have heterogeneous cells, but you could also have homogeneous. So this is a very nice example, I think, for the small things that you only learn and see when you have the chance to work with someone with lots of experience. So I think take this as a metaphor as well. I mean, not only the practical meaning of it, and take it as a message. And I think mentoring is important in every stage of your career development. So going on, what else did we do? We had a field day in a search for a charcoal production site in the nearby hill. That's a region which is very close to the lab where we worked. So it's located in a beech forest. You could see the arrow at the bottom here. This is the very nice outline of the Lake Constance. And you see more or less the location where we looked for that site. And on the top slide, you could see the area of that Schienerberg. I'm not sure if I pronounced that very well. Hill, that mountain. So here again, as you could imagine, on a field walk, you have like lots of discussions and things you could really comprehend better being outside in the forest. And as you could see, this is like predominantly beech forest, which is very, it is a species I'm getting really interested at. So this is one of the days. I mean, it wasn't all lab work. There was also a little bit of survey walk in the mountain. And also based on that, we kind of opened. Sorry? Can you hear me?
Yes, yes.
Yes,
Okay.
yes.
Good, thank you. So after the field survey, we opened a little bit of the discussion of the. I think this is not for the working group dedicated to archaeology. This is more the history working group. But we, as you see, we talked about the survey techniques and things we could see while practicing our hobbies, as you see here. So I shared a very recent, say, accidental discovery I did literally a week before I go to Hemmenhofen. I was skiing in the mountain, and in the area we were downhilling. I spotted these scars for resin collection on Pinus heldraechi. So you could see in the middle you have an older scar already covered with well, bark tissue, and then to the left and my happy face to the very right is a more recent one. I can't really say how recent is that because to my knowledge and some ethnographical reading I've done, I mean, that practice in that particular mountain has posed for at least 50 years. So this is another chapter we discussed and talked about. And basically, this is it. Because I visited during Easter, so the south of Germany during the Easter time is very pretty. You see these flowering magnolia trees. And this is my thank you for your attention. And I'm here for questions if you have some.
Thank you very much for this very interesting and synthetic presentation, Mila. Thank you very much. And now we can start discussion. If you have some questions, if you have some doubts or you need some clarification, now we have time for this.
Also, now I'm stopping the recording.
Thank you.
You're welcome.